Hello everyone, my name is Andreas and I work as a postdoctoral researcher at the University of Edinburgh. And today I will present you Mobius Assembly. Mobius Assembly is a fast and user-friendly cloning system that I developed during my PhD. As you probably know, standardization is an important principle for synthetic biology because it allows different labs to exchange protocols, methods, parts, and this can accelerate biology. The first attempt to introduce standards in plasmid assembly was done in 1996 by Rembachuk with the development of NOMAD. NOMAD was using type 2S restriction enzymes to make standardized building blocks and construct reusable modules. However, even though it was a really good attempt for standardization, it was not recognized by the broad scientific community. Type 2S restriction enzymes, it's a category of restriction enzymes that they recognize asymmetric DNA sequences and they cut outside the recognition sequence. For example, here we have BSA1, and on the left highlighted you can see the recognition sequence, and next to the recognition sequence we have the cutting sequence. 10 years later, we have the development of Golden Gate assembly, which again, it uses type 2S restriction enzymes. The restriction sites are eliminated from the ligate product, so digestion and ligation can happen at the same time. And the user can set the overhangs, which allows for unidirectional and scarless assembly. In this example, we have the combination of a promoter, a coding sequence, and a terminator. There is a release of the parts with the action of a type 2S restriction enzyme. And then with the action of the T4 ligase, we have the assembly of the parts in an acceptor vector in a one-tube reaction. Even though Golden Gate assembly was a powerful cloning method, it doesn't allow for the reusability of the composite parts. And this was changed with the development of Moklo and Golden Bread in 2011, which they introduced a second type 2S restriction enzyme, allowing the construction of more complicated constructs. In general, Moklo is characterized by high cloning capacity, but it has a complex vector toolkit. And on the other hand, Golden Bread, it has a simple vector toolkit, but low cloning capacity. Over the years, we have the development of several Golden Gate variants, which they try to tackle some problems of the Golden Gate assembly, and also to adopt the Golden Gate cloning in different organisms. Most of them, they follow the Moklo philosophy, and in general, they have, again, high cloning capacity and a complex vector toolkit. Because I wanted to combine benefits of both Moklo and Golden Bread, I developed Mobius Assembly, which combines high cloning capacity with vector simplicity. A bit later, we also have the development of loop assembly from another group, which also shared this philosophy. So Mobius assembly cloning is based on a two-level approach for transcriptional unit and multi-transcriptional unit augmentation. As I said before, it combines high cloning capacity with a simple vector toolkit. It has lower domestication requirements. Domestication is the removal of the internal type 2S restriction recognition sites. from the sequence. I use a visual cloning screening by chromogenic proteins, and Mobius Assembly is direct compatible with existing part libraries. So in level 0, which is the storage level, we have just one vector, the Mobius universal acceptor vector. In level 1, we have 4 acceptor vectors, as well as in level 2, we have again 4 acceptor vectors. We also have 7 auxiliary plasmids, which they help in level 2 cloning. So from level 0, we go to level 1 and we create a transcriptional unit. From level 1, we go to level 2 and we can make up to 4 multi-transcriptional units. And then if we go back from level 2 to level 1 again, we can make up to 16 transcriptional units. And we can go back and forth by the 2 levels, and we can increase the number of transcriptional units by following the geometric sequence 1, 4, 16, 64. Mobius Assembly uses chromoproteins to replace the blue-white screening, and the use of chromoproteins eliminates the need to use IPTG and XCAL in the plates, which make the cloning less costly. And also, it is easier to distinguish between the different cloning levels. In the universal acceptor vector, I'm using AmylCP, which gives this purple color. All the level 1 acceptor vectors has space pink, which gives pink color. And in level 2, we have the expression of superfolder GFP, which gives this yellow color. When we have successful cloning, the colony color changes to white. So in level 0, in level 0 we have we convert the sequences into standard parts. So we amplify or we synthesize a sequence which has the proper overhangs, and we combine it with the universal acceptor vector that results to the creation of a Python brick. Level 0 cloning is done by the use of AR1 restriction enzyme, which is a rare cutter, and as I said before, it reduces domestication requirements. Generally, the enzymes that they are being used in Golden Gate, they have 6 base pairs recognition sequence, but AR1 has 7. And I forgot to mention that overhangs are set according to the Python brick standard. So the Python brick standard are 4 base pairs overhangs that they flank the standard parts. We have an overhang on the left and an overhang on the right of the sequence. And there are different, as you see on this image, there are different overhangs for the different parts. Python brick standard, it follows by different labs, and it facilitates part sharing. In level 1 assembly, we have the combination of standard parts into transcriptional units. The cloning level 1 is done by BSA1 restriction enzyme. And in this example, we have 3 Python bricks, 1 promoter, 1 gene, and 1 terminator, which they are combined together in a level 1 acceptor vector, and they give 1 transcriptional unit. Level 2 assembly, we have the generation of multi-transcriptional units. In this example, we have the transcriptional unit 1, transcriptional unit 2, 3, and 4, along with the auxiliary plasmid 4a, which are combined together in a level 2 acceptor vector, and they give a 4 transcriptional unit construct. Cloning in level 2 is done by AR1 restriction enzyme again. So, how do we use the auxiliary plasmids? We have in total 7 auxiliary plasmids. The first 3, they provide middle to end linkers, which are being used when we assemble constructs up to 3 transitional units. On the other hand, we use the auxiliary plasmids for alpha, beta, gamma, and delta when we assemble 4 transitional units together in the 4 different acceptor vectors, alpha, beta, and gamma. In this example, level 2a acceptor vector, we want to assemble 4 transitional units, 1, 2, 3, 4, and for this reason we use the auxiliary plasmid 4a, 4 transitional units, and in the acceptor vector level 2a. In this case, where we use the level 2 acceptor vector b, we want to assemble 3 transitional units, 5, 6, and 7. And for this reason, we use the auxiliary plasmid 3. And then again, from level 2 assembly, we can go back to level 1 assembly. For example, this time we have 4 transitional units in the level 2a vector, 4 transitional units in the level 2 beta vector, 4 in the 2 gamma, and 4 in the 2 delta vector. And we combine all of this together again in a level 1 acceptor vector, and we get a 16 transitional unit construct. As I said before, cloning in level 1 assembly is being done with the use of the BSA1 restriction enzyme. And in level 1 assembly, we never use auxiliary plasmids. To prove the functionality of the cloning system, I reconstruct the violacein operon. Violacein, it's a chromogenic molecule with antibiotic properties, which is produced by the Chromobacterium violaceum, and it converts, the operon converts the L-tryptophan to violacein with the action of the enzymes VIO-A, VIO-B, VIO-E, VIO-D, and VIO-C. In this example, we combine the genes VIO-A, B, D, and E in a level 2 acceptor vector A, and we got black colonies because of the production of the protoviolaceinic acid. Next, we proceed to the assembly of carotenoid transitional units. In the first example, we combine CRT E, B, and I in a level 2 acceptor vector A, and we got pink colonies because of the production of lycopene. Then we combine CRT E, B, I, and Y in a level 2 acceptor vector A, and we got orange colonies because of the production of beta carotene. And then in a level 2 acceptor vector beta, we clone the gene CRTZ. In the next step, we combine this transcriptional unit CRTEBIY with CRTZ in a level 1 acceptor vector A, and and we got the construct CRT E, B, I, Y, and Z, which resulted in the production of zeaxanthin, which gives yellow color in the colonies. Finally, to prove the high capacity of Mobius assembly, we proceed to the construction of a 16 transcriptional unit construct comprised of carotenoids, chromoproteins, and violacine genes. First, we clone the genes CRTE, B, I, and Y in level 2 acceptor vector A. The chromoproteins SC orange, amyl GFP, AMAG lime, and FW yellow in level 2 acceptor vector B. The red chromoproteins TS purple, FO red, ASCP, MRFP1 in the acceptor vector level 2 acceptor vector gamma, and finally in level 2 acceptor vector delta, the violacine genes. As you can see from the plates below, the first construct resulted in orange color because of the production of beta carotene. The second construct resulted in yellow colonies because we have combination of yellow chromoproteins. The third construct resulted in pinkish red colonies because of the combination of red colors. And lastly, in the fourth plate, we have again black colonies because of the production of protoviolacinic acid. In a second step, we combine all of these four vectors with the genes back to a level 1 acceptor vector, and we make this big construct, 16 transcriptional unit construct, comprised of carotenoids, chromoproteins, and violacine. When we transform the construct to E. coli cells, the colonies got this black color because the violacine production was dominant to the other colors. And also verification of the construct was done by restriction digestion, showing the correct pattern on agar gel. And then, apart from E. coli, we adjusted Mobius assembly to be used in plants. We generate a new acceptor vector for plant expression, which has reduced plasmid size and bears a new origin of replication in E. coli and Agrobacterium. And also, we clone and characterize a library of standard parts for plant expression. To sum up, I developed a Mobius assembly which has reduced domestication requirements. It has a minimal vector toolkit, high cloning capacity. It follows the phytobrick standard. There is no need to use IPTG or XGAL for selection. Vectors, there are currently available vectors for E. coli and plants, and is directly compatible with several Golden Gate libraries. If you need more information about Mobius assembly, you can read the paper. And also, there are information about the parts in AdGen. Thank you very much for listening. And if you have any question, you can drop me an email.